Hi everybody, we're going to work on completing your graphic organizer for simple organisms, specifically working today on viruses and bacteria. This is on page 12A of your handbook. So we'll start by drawing a simple picture of a virus. I do need you to sketch this virus in the space provided, and I need you to label each of the components of the virus. Now, as you're doing so, notice that this virus does not have a cellular structure. It does not have a cell membrane. It does not have a nucleus. It doesn't have all of the organelles that we typically find in cells. Viruses are not made of cells. But do notice as you are drawing it that inside that protein shell, inside that capsid, there is a nucleic acid. This nucleic acid might be DNA or it might be RNA. So although it does have that component of what a living thing might have, it is not a cellular structure. It does not have a cell membrane. Instead, it's surrounded by a protein capsid, and therefore we do not consider viruses to be alive. Viruses, like living things, can reproduce. However, they must have a host cell in order to do so. So in the space provided on your graphic organizer, I need you to write down the information about the lytic cycle and the lysogenic cycle, which are the two viral cycles of reproduction. Now there is a diagram, the same diagram that you see here on your graphic organizer, showing the difference between those two cycles. So look carefully at that diagram and notice that the virus, which is labeled bacteriophage, this is a virus that actually infects a bacterium, has attached to the cell. You can see the DNA inside the virus. It looks like an S. If you look at the next image, you will see that the DNA has been injected into the bacterial cell. So that DNA is going to use that virus DNA is going to use the bacteria, the host cell's DNA, to make copies of itself. So if you follow the arrow up, you will see in that next picture that new virus parts are being created. Then those virus parts are going to be assembled. And then finally, if you are looking at the top line, the last picture, you will see that those new viruses are leaving the host cell. This does kill the host cell. And each of those new viruses can go make more copies using new host cells. But then going back down to the bottom, we have the virus attaching to the host cell, injecting its DNA into the host cell. But then look down at the bottom, you will see that the DNA is actually becoming part of the host cell's DNA. So it's going to stay in there dormant for a while. And that's what you see in the last picture. It actually becomes part of the host cell. And so at any point, something can trigger that viral DNA to pop out and start making copies of itself and go through that lytic cycle of reproduction. So viruses that use the lysogenic cycle of reproduction are those that you always have. You never get rid of them. They are always part of the host cell's DNA, like the herpes virus, for example. You never get rid of that. You may not always have symptoms, but if you have herpes, you always have herpes. Now, viruses can be prevented. You get a vaccination to prevent viruses. When you get a vaccination like a flu shot, the doctor is actually using a dead or weakened form of the virus and injecting that into your bloodstream. And what that does then is activates your immune response, your body's own medicine. Your body is able to produce antibodies, which is a different thing from antibiotics. These antibiotics bodies are like your body's own medicine to defend it against these viruses. So by putting a little bit of the virus into your bloodstream, the virus actually turns on your immune system. So getting immunity to a virus in this way is called active immunity. You went and got a flu shot, so you have active immunity to the flu because your body has produced antibodies against the flu. Same thing goes for if you get a virus like chickenpox. If you get the chickenpox, your body has active immunity, and next time you come into contact with it, it will turn on that immune response, and you probably won't get the symptoms of chickenpox. Passive immunity happens when these antibodies are passed from one person to another, and this is most commonly done through breast milk. This is why new mothers are encouraged to breastfeed their babies, because babies haven't built up any of their own active immunity. But if the mother breastfeeds, some of the antibodies from the mother can be passed into the child and give them temporary immunity to some of the viruses and other pathogens that they might encounter. Now, an antibiotic is a whole different thing. An antibiotic is a medication that will not kill a virus. An antibiotic attacks the cell wall of an organism, and you just learned that viruses aren't cells and they don't have cell walls. So an antibiotic does not help a virus at all. Antibiotics are used against bacterial infections. You may remember that year after year after year, you have to get new vaccinations for the flu because viruses, through natural selection, change every year. They mutate. Remember, mutations are the raw materials for natural selection. And those that are selected for, they have favorable traits, survive. These are the viruses. They survive, they reproduce, and they pass those traits on. That's natural selection. And so every year the flu virus is a little bit different. And so there is a new vaccination to protect people against the flu virus. So hopefully you filled in that section on your graphic organizer. We'll take a quick look at bacteria next. The place to draw the bacteria cell is quite small. I'll tell you the things that you need to label after you have sketched the bacterial cell. You need to label the cell wall. So look carefully at the picture and make sure you label it correctly. You need to label the chromosome, which on this picture is labeled nucleoid. It's all of that stringy stuff, the stringy DNA in the middle. And you need to label the ribosomes, which are the tiny little dots. And then finally the flagella, which is that whip-like tail. Bacteria are living things. They are only one cell big, unicellular. They do not have a nucleus. Pro is no. They are prokaryotic. They do contain DNA in the form of a big chromosome and in plasmids, as well as ribosomes, and are surrounded by a cell wall. So make sure you've drawn that diagram. Bacteria can also reproduce. Their reproduction is asexual. This is accomplished through binary fission. You learned about that in Module 5. In binary fission, you have one single cell that splits into two equally sized cells, so it makes a clone. However, bacteria can exchange DNA, plasmid DNA, through tiny little string-like structures called pili. And when they exchange DNA, this allows for genetic variation. So it's mixing up the DNA in a sense. And so there you can see two bacteria that are connected by that pili, and little tiny rings of DNA, plasmids, can pass through that, mixing up the DNA and allowing for genetic variation. There are bacteria that are bad. These are called pathogenic bacteria. These pathogenic bacteria are killed by antibiotics. An example of a bacterial infection that can be killed by antibiotics is tuberculosis bacteria. But there are far more good bacteria. For example, there are bacteria that decompose wastes. There are bacteria that help us to digest food.